them, but still to set the stage up. So for me, active particle is something that can move by themselves without the application of any external force pushing or pulling it. Okay, that's the working definition for my talk. That's an active particle. Okay, here is an example. It's a micro swimmer. The size is micron size. It's in fluid and it's swimming. This is another example. Is from our simulations. It's a slightly complicated micro swimmer with an elongated sail body and the propellers that you can see here. There are multiple ones. Okay, so either both of them actually swim in the same way. So the sail body is sort of a rigid structure, and then you have a flagella which is flexible. The flagella, this is propelling part, that pushes the fluid backwards. Pretty much like when you swim, we push the fluid backwards with our leg, and as a result there is a thrust force that is generated that acts on the sail body, and because of that the whole thing moves forward. So you can have some other sorts where the propellers are in front of the body, so they kind of pull the fluid inwards, and as a result they move. Okay, so this is they pull the fluid back this way, that therefore the thrust force is again that way, so the whole thing moves in this way. So whenever these objects move in fluid, so they will create disturbances, and so the flow patterns are like that, for example, and then when you bring in another particle nearby, another swimmer, so they will interact via these disturbances. Okay, those interactions are called the hydrodynamic interaction, and this is what we are going to talk about mostly. Okay, so there are two types of swimmers, although we are not going to need it, but in general there are two basic types of swimmers. So this swimmer pushes the fluid both in the forward and the backward direction, that's why it's called a pusher. This, on the other hand, pulls the fluid inwards both in the forward and the backward direction, that's why it's called a puller. Okay, so there are a large class of problems of active particles where these interactions are very, very important. Hydrodynamic interactions are very important, very rich dynamics, and they play huge big roles in the phenomenology that is observed, and that's why so the task to include them in those kind of problems is looked for, and this is the first step one can take. So the idea is to calculate the disturbance around swimmer so that you can put another swimmer and then find what is the interaction. So to find the disturbance around the swimmer, you have to look at the effect of these tiny forces that the flagella puts into the fluid, also how the sail body is pushing forward into the fluid, and then see what is the effect on the fluid. The minimal model to do that is the following. So you just assume that all the forces that the flagella is putting on the fluid, there is just a point force, this point force, okay, and then when the swimmer moves this way, the sail body puts another force, and that's this point force. Okay, you just model the disturbances by this, just by these 2 point forces, and then the requirement is that these 2 point forces should cancel each other. They are equal and opposite because there is no net force here that pulls the object. It actually moves by itself, right? So therefore, this is the minimum requirement. You just need 2 point forces, equal and opposite, and then you put that into the fluid, then the disturbance created by this object when you are far away from it, and the disturbance created by these 2 point forces when you are far away from it, they are similar. Okay, that's the first assumption, first step that you can take. Turns out that it's a minimal model, but it's a powerful model. Okay, for example, in active matter, an active stress is created just because of the swimming, and the form of the active stress is like that. Suppose you have an ordered state like this, so you have elongated particles, these are the swimmers, and they are, for example, oriented in one particular direction that is denoted by n locally. Then the swimming activity will put stresses which are called active stresses that can be written down like that. And to arrive at this expression, you basically assume that the swimmers are stresslets like this. These are called stresslets. Okay, so already with this form is included in many hydrodynamic theories, and then lots of theoretical predictions have been made, and so this is a big success. Okay. So the point is that the minimal state is powerful. However, I mean, this talk is about situations where that is not enough. Okay. So we are going to talk about 3 different scenarios, I mean, if time permits. Okay. The first step, first scenario is that, I mean, when you have a big dense suspension of these micro swimmers, then as I already said, the disturbances are similar only when you are far away from the object, far away from the swimmer. So when a dense suspension is there, so the swimmers are close by, therefore you expect that the minimal model is not enough. However, surprisingly, I mean, if you even take one swimmer and put that in an imposed flow, you will see that the minimal model is not sufficient. Okay. So that's one part. The second part, I mean, I'll be talking about when we put this swimmer near a substrate. Okay. I'll argue that even there the minimal model might not be enough. And then, so we'll talk about a detailed model of the of a bacteria. Okay, so where so you will see that it's actually quite realistic model, and you can observe the motility in time scale, which are very small. Okay, compared to the time scales that we are interested there. Even in that time scale, you need hydrodynamics going beyond just the stress rate assumption. Okay, so these are the people who are involved. So Direk and Ambaresh are the PhD students in my group, and Pierre is a joint student between me and our collaborator Holger Stark at TU Berlin. Okay. Yeah. So the first problem. So by the way, so probably many of you have heard about this talk, about this part of the work. I'm sorry for repeating the same. So I'll try to go a little bit faster, maybe in some portions. But I mean, bear with me. So some parts are new here. Okay. Okay. So the first part is a dense suspension of bacteria. So the work was inspired by an observation done on a very dense suspension of E. coli, which is a kind of a particular type of bacteria, microswimmer, and this concentration is rather high. Okay. So there, in the suspension, these are the tracer particles, and it turns out that the tracer particles move in an elliptical orbit, okay, in a synchronized way. So interestingly, if you look at the trajectory of individual cells, they are still random. Okay, so they look like random, although the tracer particles still perform this elliptical motion there. That means that this comes as a collective dynamics. Okay, so collectively the hydrodynamic flows and all that. I mean, so we can see that the hydrodynamic flows will have a big impact on it. So that was just a motivation for us to, so Direk started with this model. So we introduced a model that was suitable for the dense limit, and in one particular way, that in the dense limit there is no particular reason to exclude the bending of the flagella. Okay, so this is high density, so the cell body can bend the flagella, which are fairly flexible objects. Okay, so that's what we allowed, and then this is like the bottom-up approach. So we look at cell by cell, and then we want to go to the dense limit. Okay, that was our main motivation to start with, and at the end, just now we have started to look at it also from the top-down limit. So we started with looking at the continuum limit of it. So although I'm not going to talk about it. So once we had developed the model for going beyond the stresslet, so we found that, as I mentioned earlier, even a single swimmer dynamics is quite rich when we have imposed flow. So that motivation came from this work where a bunch of these swimmers were taken. So this is this particular micro swimmer, which is fairly round and rigid object. So that was taken in a channel where a flow was imposed. So flow is coming from that side to this side, and in normal conditions, the swimmers simply follow the streamlines. So if you start with a homogeneous concentration across the channel, the concentration remains as it is, okay, for all the time. So however, the swimmers are they get attracted towards light. Okay, so they like to turn towards light. So when a light source is the light source of the correct wavelength was turned on, it is found that the swimmers still they are actually flowing along the flow, but they get collected along the center of the channel. Okay. So the explanation was given like this. I mean, so a simple explanation is that the flow here is not uniform; it's parabolic in nature. This is the channel width. Okay. So because of that, a swimmer that is on the top half of this channel, that will actually encounter a vorticity that is trying to rotate it in that way, and when a light source is turned on, this swimmer will get a torque from the light orientation and in the opposite direction, so that the swimmer will have a orientation towards the center, and likewise on the bottom half. Okay, and therefore the swimmer in presence of the light will travel towards the center. Okay, that was the explanation, hand-waving argument. So the important part here is that the cross-stream migration that you can see, the focusing here happens only when the light is present. Without the light, that doesn't happen. Okay, that was the observation. So that was theoretically understood also. This is the theory, very simple theory. So you have activity, that means that the particle swims with a particular velocity. This is the activity. It also fills the flow field, that's the flow field, and also the flow field can rotate it, that's the vorticity part. Okay, and if the particles are elongated like that, okay, they can also feel the rotation due to its elongation, the geoffrey orbit type of motion. Okay. So this has already been studied, and the result is that if you do not have any light source, the particle performs this oscillatory motion, but on an average they are kind of parallel to the center line. This is the center line of the channel. Okay. So the trajectory is on an average parallel to the center line. So even in this elongated object, this trajectory is parallel to this center plane. Okay. So that was the observation. They oscillate because the vorticity, if the swimmer is on the top half of the channel, the vorticity will rotate it, and because the swimmer is active, it has an active propulsion, so it moves in the instantaneous direction. So together it will actually perform this this oscillatory motion. Okay. So if you consider both of them together. Okay. So here what is being considered is that the objects are rigid, and physically they are head-tail symmetric. Okay. So although there is an activity direction there, it's an intrinsic direction, but physically the particles are still head-tail symmetric. So therefore, when this object move in a fluid, the flow that I showed schematically earlier will not be coupled to its own motion. Okay. So now let us look at the other example that came from our simulation, for example. So here the cell body is rigid, but it has a flexible flagella, and then for this type already you can see that the head is different from the tail. Also the flow that is created by the cell body might affect the dynamics of the flagella and vice versa. Okay. So we are asking what happens in this case. Okay. So turns out that to address that, you need to actually incorporate the hydrodynamics in a little bit more detail. So we have to go beyond this first step that I talked about. So then we reach that stage in couple of more steps. Okay, and the first of those steps is that instead of saying that this point force acting directly on the fluid, you assume that there is a cell body. Okay, like that, and then the opposite of that thrust force acts on the cell body, and the cell body moves. As it moves, this force ultimately will be transferred to the fluid, but through its surface. It's an extended surface. So basically you have introduced the cell body. So as you I mean, this is accurate when you are far away. So as you approach that swimmer, you are going to see this extended nature of the cell body and the flagella and so on. So this is the first improvement that you can do upon that. And then you can solve the problem to find the cell propulsion velocity, and then that is plotted here. So what it means is that suppose this point force is far away, then this will act like a sphere that is being dragged by an isolated force. Okay, that's the velocity of that sphere. But when this point force is, you bring it closer, the point force also creates flow field that will reduce the velocity of the swimmer itself. So that's the reduction in the velocity that you see. Okay, so this came out of the solution of the model, but once you know the cell propulsion dynamics, cell propulsion velocity, now we can calculate the disturbance that is created around this model. So that has different contributions. For example, this sphere is moving in a fluid, so that will disturb the fluid, that is that contribution. So that is written here. Okay. There is this point force which represents the flagellar force on the fluid, that will create its own disturbance, that is represented there. Okay, those are kind of the straightforward trivial ones which are also in the textbooks. However, the non-trivial part comes when we consider that this point force is not isolated totally, it is not in the bulk, but it is near the cell body. Therefore, the flow will be kind of reflected off the cell body. Okay. So that part is little bit non-trivial. However, that can be solved by using something called method of images, and that uses the fact that now what we need is that there is a point force in the vicinity of a sphere. On the surface of that, the velocity is 0. Okay. That is the stick boundary that is most often encountered in experiment. Okay. Stick boundary condition. So u is 0 here, the flow field, and there is a point force there. Then we need to find the flow field in this region. Okay. Turns out that a very similar problem exists in electrodynamics, where electrostatics, where there is a charge in front of a grounded sphere. So that potential on the ground is 0. You need to find the potential in this region. And then there is indeed a correspondence between the Stokes flow that we are solving for the fluid disturbances. Okay, this is the high viscosity limit or low Reynolds number limit of the Navier-Stokes equation that gives the dynamics of the fluid. Okay, so that is Stokes equation. At this, you can see that this object is similar to that apart from tensorial details. So turns out that, I mean, the solution of that, the green function of that is similar looking object to the green function of this solution, this equation. And that is why apart from some tensorial details, things are very similar. So the solution here was to introduce an image field. Okay. So image charge which has this magnitude, but you introduce that inside the sphere such that together they make the potential go to 0 on the surface. Okay. So once that is satisfied because of uniqueness theorem, you can see, you will find that the potential here is exactly the potential here for the original problem is exactly the potential created by this object and that object. Okay. The same solution exists for this situation. So the image of the point force is also located at a very similar looking point. Okay. However, the image is not a single object, but they are a combination of 3 singularities. Each of them will create different patterns of flow. So now if you combine these 3 flows, then you will get this disturbance. Then you add all these 3 contributions, then you get the disturbance, total disturbance around the swimmer. Okay. So with that, you can get the streamlines, you can get another swimmer nearby, and you can calculate the hydrodynamic interaction and so on at that level. Okay. So this we did. So that was the first modification we did, and then the point was that in the dense suspension we said that the bacteria can collide with the flagella of another bacteria. So we needed a physical flagella, not just a point force, so that another bacteria come and collide and bend it. Okay. So for that we wanted to introduce the model a little bit. So one step by one step. The next modification is that if the flagella is bent, so then the earlier point force was actually so like that, now it will be like this. So what happens is that this model, so the non-bent model was an axisymmetric model, this is a non-axisymmetric model, that is the first complicacy. But luckily this can be decomposed into 3 parts, a purely axisymmetric part, a purely non-axisymmetric part, and an active rotation. So solving for this is non-trivial again, but we can still do it, and after doing that, this is the flow field that we get as a function of the bend. Okay. So I am skipping these results, but then comes the final step for that part of the work is that we need to introduce the physical flagella. Okay. So that is what we did with this set of equations. You don't have to understand all of them, so I am not going to explain fully, but in principle what we are doing is that this flagella is pushing the fluid at various points along its length. Okay. This represents the flagella of the real swimmer. So that creates flow field. This is a slender body, it is a very thin object, but when it moves, it will also create disturbances in the fluid. That is also included. So, so these are, for example, the forces and the forces on the cell body and on the rod. So rod is a slender body, that's why you see the integration here. These are similarly torques on those objects, and these blue terms are the flows on the background. Okay. So they are very messy looking terms, but they basically represents the flow that is experienced by each part of different parts of the object. Okay. So now there can be interaction between this flow created by the cell body with the dynamics of the flagella and vice versa. On top of it, so if there is a bend, then we introduce a torque, that restoring torque that tries to restore the original orientation. So there is a spring constant K. If K is small, then this is quite flexible, it can actually flip quite easily. If it is large, then it is rigid. Okay. So apart from that, we have other conditions like force balance, and then when we introduce a flow, the flow itself gets distorted because of the extended nature and so on. All these things are included in the model. So once you do that, by the way, because of the non-axisymmetric nature, you see that the image of each of these point forces, earlier it was just a point image, but here it becomes extended. Okay. So you can see the complicacy that arises when you make things that much complicated. I mean, you deviate from minimal model, but the outcome you can see. Now this is the flow field. Okay. You see it's drastically different, right? Now, so what once you have this model, you put in a imposed flow. We said that, I mean, so for dense suspensions it might be important, but what happens if we have an imposed flow? That's the question we ask, and this is the result we get. These are the trajectories of individual swimmers, okay, along the channel length. So this is the channel length, this is the channel width. Okay. Already you can see there is no light source. Okay, there is no external orienting field, but still the swimmers are moving away from the center. That is the new thing that you get once you include the complete dynamics, and it's qualitatively different, not just quantitatively. Okay. So there are three different population. If the swimmer starts well above the center line, they will go to that respective wall. If they are little bit towards the center, they will start oscillating first and then ultimately pick up one of the directions and move towards that wall. However, if they start very close to the center, so they are kind of trapped to the center. Okay, so most of the population will go to this wall. Only a very tiny population will stay near the wall, center. Okay, so this is the observation that so there is a cross stream migration even without any external orienting field. Okay, so this is yeah. So important thing, we analyze why it happens. So for example, if you have asymmetric particle, head tail asymmetric particle, but they are not swimming, then you don't see this cross stream migration. If you have active particles but they are symmetric, like the one that I showed earlier in the earlier examples experiments, there also you don't see it. The key aspect is these two, activity plus asymmetry. Asymmetry meaning head tail asymmetry, which means that you have to go higher order in hydrodynamics. Okay, you have to include the extended nature of the cell body. Okay, so we analyze that theoretically. So I mean, we can find the angular velocity as the swimmer tumbling and moving like this. We can find the angular velocity as a function of theta, and we see that when the swimmer is pointing in the vertically up direction, the angular velocity is slightly less. This asymmetry is resulting from the head tail asymmetry. That means that when it is pointing upwards, it spends slightly more time than when it is pointing downwards as it tumbles. Okay, that is the reason why the trajectory is bending upwards in time. Okay, so this is from the particular asymptotic limit of the theoretical equations. This is from the full simulations, and from the full simulations also we can find the same curve, and then we can see that they are matching with that explanation, meaning that again you need to go beyond just this stress-led model. Okay, so not only that, there, I mean, if you dig more into it, you will see that rich dynamics is there. Okay. Right, yeah.
There should be flexibility and it should have its own mode.
Right. So that is the third part of the talk. So we have a model, but probably I will not be able to explain. But you can do that. Okay, there you can actually probe the system even at a shorter time scale. Okay, so we are ignoring that kind of fluctuations. Okay, yeah. Okay, so I mean you can probe for dynamics, and so because of time constraint, I am not going fully into it. What time my talk ends? Sorry? I have 7 more minutes. Okay. So then I am skipping the details here. So, okay. So, I mean just two things, that with this inclusion of the detailed hydrodynamics, we can predict some controls over the system. For example, we can start playing around with certain values. So, for example, the shear rate, so that the velocity at the center, for example, of the imposed flow, you can play with that, and just by playing with that, you can actually increase the population that gets trapped in the center. Okay. Also, if you introduce a aligning field, you can focus the particles at a particular height. So earlier they were actually focused only at the center, but now you can focus them at any height that you want by adjusting the angle of the orienting light, and then you can think of creating certain devices where you can get particle currents in one situation and no currents in other situation, which is like acting like a living transistor made up of particle currents, swimmer currents. Okay, so just for I mean because of the lack of time, so I am moving directly to this slide. So if we have this model, so even the full model, okay, so with a rigid cell body and a rigid rod, and you zoom out of it, then the flow field looks like this. Okay, so if even it happens, this is from between -500 to 500. So this is the length is in terms of the radius of the cell body. Okay, but the stressless field, this is the field that is created by 2 point forces. Okay, so that is I mean you can see that then if you are actually interested in this kind of concentration, then stressless is good enough. Okay. However, now look at the situation where the swimmer is non-axisymmetric. So this is at -1000 to +1000. This is even further. Okay, -1500. You can see that the swirling is still there. Okay, this is not unlike that. So that means that if you want to capture the collective dynamics of these objects at that length scale, stressless is not enough. Okay, so this is the stressless approximation is not good for non-axisymmetric swimmers. One more thing is that this is the hurdle that we faced. Okay, so when we are developing, so we are actually following this reference, we model as a sphere rod assembly, and where we neglected the flow field created by the movement of these objects. So we thought that the rod is very thin, and when it moves, it will not create sufficient amount of flow. But if you do that, then then plot the flow field, then that looks very different from the stressless. Okay, it looks like as if there is a point force somewhere, and that actually fundamentally is wrong, because there should not be any external force. So you have to go to that limit. Actually, there is no way. Okay, we cannot get around that. So with that, I mean we are now going into the study of collective dynamics. Okay, we have also started to look at in the continuum limit. Okay, so the other problem. Okay, I'm just going quickly now. Sorry for that. Other problem is that just put the same swimmer near a wall. Okay, then see what happens. Okay, again the problem is very, very complicated now, because now you have to satisfy the boundary condition not only on the cell body but also on the wall simultaneously. That makes the problem very, very hard. So and also so earlier this point force had an image here inside this sphere, but every one of those singularities will have an image down the wall as well. So and these images are not just single objects, they are like set of singularities. So all these you include, and then so these are some of the examples. Those are the flow field created by these singularities in the bulk. Those are the flow field near the wall. Okay, so this is how the flow field would vary if you consider this type of swimmers, where the point force here is distributed in different points. Okay, but still you do not have a physical flagella. Okay, this is how the flow field will vary as a function of angle orientation. Okay, so why we are interested in it? So the one reason is that, I mean, so this is this is from an experiment. So and this is the bacteria called E. coli. It is released from a particular height on a substrate. The bacteria moves toward the substrate and then it gets trapped. So it actually keeps on moving along the substrate. Okay, it cannot come out of it. Okay, this phenomena is called hydrodynamic trapping. It is important for various processes, even they have very good biotechnological or medical applications, but that process in physical terms is not fully understood. Okay, that's one reason. Second reason is that, I mean, there is a paper from observation is that you take a drop of fluid between two plates and you put bacteria in it, then the bacteria gets collected on the two substrates. Okay, so this is the concentration profile along the width here. Okay, that is why that's width is this. Okay, you can see that the bacteria concentration is high near the substrates. Okay, so those dots are from experiment and the solid lines are from theory, and the theory explains this as follows. So each of the swimmer is a stresslet. Okay, this is a real bacteria, and they said that they can be approximated by a stresslet, the minimal model that I talked about. The stresslet will interact with the wall via these image fields. Okay, as a result, their orientation will be parallel. Okay, if you consider a stresslet, then the stresslet will be parallel to the wall. Once it is parallel, the flow field that is created by these will be downwards. So the swimmer first gets parallel, and then once they get parallel, because they are stresslet and because their reflection, the reflections are actually downwards, so it will get attracted towards the wall. Okay, so that is the theory, and then we sort of try to understand with our model. Okay, so what are the models that at our disposal? As I said, we can have this model because in the bulk it has the correct stresslet field when you are far away from it, and also you have the full model who have the sphere rod assembly. Okay, the other model where you neglect the flow due to the rod is not correct. Okay, like I said earlier. So therefore, only these two models are there, but can we actually work on this minimal model, because this is very, very difficult to solve. Okay, really difficult. Okay, even the streamlines takes kind of half an hour to 1 hour to generate. Okay, forget about simulating. So we tried that. Okay, we tried this one first. Okay, let's see what happens. So this is, I mean, again, lack of time. I'm just physically showing what's happening. So if you take that swimmer, this kind of swimmer, and this is the wall, for example. So if you are far away, I mean really far away, then you can see that they are actually parallelly oriented, like the experiment, like the stresslet example. However, you have to go that far where diffusion, rotational diffusion dominates over hydrodynamic interaction. So that orientation doesn't mean anything because diffusion dominates. So this is the height where diffusion dominates, but that's where this model predicts that the model will be so normally oriented towards the wall. Okay, not parallel like that theory, like the stresslet predicted. So very close to the wall, it will assume a orientation, some orientation. So this is a wall entrapment. That means that if the swimmer comes close enough, it gets oriented towards the wall. So then it will go towards the wall directly. But the problem with that is that it actually overestimates everything because it's actively going down. So earlier it was parallel all the way, and only the fluid flow that was attracting. Here because of activity it will actually go towards directly. Okay, that will overestimate everything. So then we tested the other model, which is the full model, and we saw that now we don't have to go far. Okay, even at this height it becomes parallel. I mean, even it's slightly smaller height also, depending upon its swimmer velocity. So this is the reason where hydrodynamics still works. Okay, and then of course near the wall it gets a orientation. That means that once it reaches the wall, it actually gets trapped. Okay, because of activity. So this is kind of the explanation of the earlier movie. Okay. And then we found that angle, the steady state angle, as a function of height, and we can see that it doesn't vary much from pi, so it's more or less pi. Okay, so that means that both the experiment that I showed might be well explained by this model, but now we have to go to this model. The other model doesn't work. Okay, the minimal model doesn't work, even with a point force thing. But what's beyond, you can plot the active velocity, downward velocity, and you can see that this is quite different from these models. Okay, the full model is this. This is what is predicted by Stresslet, but it says that if you are slightly away from the wall, this is where the wall is. If you are slightly away from the wall, you are going to experience a greater attractive force. So this implies that the real bacteria are better and more strongly entrapped to the wall than predicted by Stresslet. Okay. Okay. So then, because of lack of time, I'm just finishing it here. So this is a detailed model which I am not going to talk about now, but we are also seeing these kind of detailed things which are very, very important in the time scale, where so we can see the flagellar flexibility and all that, like Pinagita asked. Yeah.
Okay. Yeah. Nice talk. Questions? Yeah, Sumesh.
So in the case where you showed in the imposed flow, right, in the post flow where you had the cross migration.
Right.
So is that because of the asymmetry coming from the finite size versus point size, or is it because of the hydrodynamic interaction between these two forces? Do you understand what I'm asking? So, you have a finite size sphere followed by a point force, and that would sample vorticity differently across streamlines, right?
So from our analysis, that we had the simplified theoretical analysis, which is basically the one asymptotic limit of our equations. This shows that the head-tail asymmetry is the reason. So suppose you have two spherical spheres connected by a rod, which creates this similar type of self-propulsion, we will not see that asymmetry. Okay, the head-tail asymmetry is the real reason. So extended nature is again a improvement over the Stresslet model.
So I have a couple of questions. One is, so when you have these two particles with just the, 2-point model with the body accounted for, and the second one is still a point.
Yeah.
So if you assume it to be, let's say, a slender structure, the Green's function is like one logarithmic, right? Instead of 1 over r. Is that something that you account for?
So you are talking about the Stresslet, right?
Yeah.
So it is 1 over r square.
No, with the head accounted for.
Head accounted for.
Like the body is accounted, but the tail is still a point.
It is still large length scale. It is still a Stresslet. Is 1 over r square.
I see.
Okay.
Okay.
Yeah. So that has to be. Okay. Because large.
Because you are saying in the next level you are replacing it with a slender object, right? And the slender object actually has logarithmic and long lengths, but I'm not talking about the stress itself, but the velocity goes as 1 over r in 3D and then logarithmic in 2D, right?
Oh yeah, so this is all in 3D, okay, so this is 1 over r square, it has to be 1 over r square large length scale, okay. Even for the correct model with the rod.
Okay, so and the second question I had was at the point of contact of the slender object and the head, is there anything interesting there?
Yeah,
how do you account
so where the model, where the flagella can fluctuate like that, so there the point of contact has to be properly accounted for. So all torques and forces are actually balanced considering that point. Okay.
I see.
Torques, so that it has to be a torque-free object because there is no external torque as well, right? So torque has to be balanced about that point. So the mechanics of that is a little bit non-trivial.
Okay, Ignacio and then Dilip.
It is related to this last slide, but to which extent the flagella really beats in a plane or rotates? So that the question is whether it's how relevant is deflection that you have accounted with respect to the fact that you can have like two counter-rotating particles, and that would lead to other type of hydrodynamic coupling.
Yeah, so the fact that the flagella pushes the fluid backwards is because of this rotation. Right, and then there has to be counter rotation of the cell body. So that means that we have to also include the rotlets into our picture. That we have done. I did not show the result. about the bend of the flagella, people have not systematically studied. But if you look at the classic papers from Berg's lab, if you look at the snapshot, then you will see those bends in places where like there are obstacles or high density.
Hi Tapan, nice talk.
Hi, thanks.
I just missed that point. The bacteria feels the presence of the wall when the distance is 100 A. What is that A parameter there?
Oh, okay. No, that depends upon lots of parameters. So it depends upon the Peclet number or the self-propulsion velocity. So the self-propulsion velocity that my student used for this part, for this particular snapshot, okay, that was 100 A. A is the radius of the cell body. Okay, but you can actually do a simple dimensional analysis type of calculation to find that height, in which rotational diffusion will be comparable to the hydrodynamic rotation.
Is the situation will be any different if you have two walls, let's say separated by 100 A, or the?
So yeah. So right now in that experiment we had two walls, but they even in that theory also assumed that the one wall, image of one wall doesn't affect the image of the other one. They are well separated in that experiment. So like hundreds or 200 or 500 microns separate. Okay. So but if they are close by, then it will be different. The dynamics will be different because the wall will talk to each other.
Okay, thank you.
Okay, let's thank Tapan for a very nice talk.
Thanks.
So we come to the last talk of this year's meeting, and we are glad that Ignacio agreed to give the talk in spite of his busy schedule. So Ignacio will also talk about active materials and, if I remember, on control. Yeah, we're good to go.
I think I'm good, yeah. Okay. Yes, here. Yeah, okay. Well, thank you for the invitation. It's really nice to be here. Yes, now. Sorry I couldn't make it yesterday, but yeah, today has been really nice day. So I'm the last one. So I also wanted to, I hadn't thought about it, but I can start with a disclaimer as because also the title, I think it points more towards where I plan to go rather than what I will really be able to cover in the presentation. So it's about active matter. So I guess being the last speaker, I don't need to motivate a lot why active systems are important, how we get inspired by nature at all scales from birds down to motors. But then we also know how to produce active systems in the lab. And I mean, I will be mostly talking about colloids. So these are a particular experimental setup of Quinker colloids in which by an applied oscillating electric field, then you can generate an instability, and then they can move together in these very well organized structures. But also there are quite a number of interesting issues related to how we can control and produce well-defined collective outcome and tasks out of local rules. These are small robots that talk to each other, but we had the first talk today was also about robots talking to each other. So this is also, I think, a very quickly growing area where many of the ideas of what I will be talking also will be relevant. As I said, at the colloidal scale, then we know in equilibrium how particles interact. When they are out of equilibrium, the type of interactions, the effective interactions, are qualitatively different. And as a result, that opens the possibility to generate, manipulate them into structures that are not possible in equilibrium. And I think that's really a new opportunity in this type of systems. And in a way, I will be touching on this part also a little bit with the hope of understanding more clearly how they interact dynamically, and not only which phases they can organize, but how they can move in response to this situation, and how, in which sense the collective response is different from the individual one. And I mean, this is just to motivate that these are a nice examples. If you put a particle in a active, in this case it's a liquid crystal, you can generate rotation, or you can also bring colloids together, create ensembles that then you can, these were done with, these are magnetic colloids with which under also an oscillating magnetic field, they can organize and then they can be moved and manipulated in a microfluidic device. All of this, if you want, within the potential that it has beyond the fundamental understanding, I think it's really what it came in this the title of the talk. We want to talk about robots, about small robots, and the idea is then if we miniaturize how we can really use them, and actually there is a part of it that I don't know, it's more science fiction. I think there is this classic by now, this Fantastic Voyage, where they pretended that it was possible to miniaturize people or have miniaturized ships going through the blood vessels, into the blood. So there is really a question of how we miniaturize. It's still going on. I mean, I was looking around, and you find when they talk about how we can use nanoparticles to cure cancer, in some places they come up with this type of images, and you may wonder whether this is real DNA with an object like this in the nanoscale, whether that's possible, or what is this about. So in reality, though, there are quite a number of experimental realizations of how to create at the scale objects that can propel or can be manipulated. For example, these are particles manipulated with ultrasounds, or these ones are particles in which these are colloids that basically decompose oxygen peroxide and generate bubbles. The oxygen peroxide decomposes in oxygen. If the oxygen, because if it's sufficiently at high production rate, it is more like a jet, and that's really then a microjet. And even at smaller scales, I mean, we have been seeing how liposomes are used to drug delivery, so now we are moving towards the nanoscale, and by also combining them with enzymes, it's possible also to make them propel. So I think that's an interesting potential, the possibility to really control the motion and the deposition of matter at small scales. As one goes from the micron towards the nanoscale, there are serious fundamental problems. Noise becomes very relevant, and as a result, a single of these objects can essentially behave as an active particle, but together can be much more sensitive to local gradients, and understanding how they react collectively and how that can affect how they can rectify motion is something that is qualitatively different to what may happen on the micron or supermicron scale, where the behavior can be more understood at the single particle level. So that was a bit what I thought for the title, but then this requires really understanding first mechanisms, and that's a bit what I will be showing, how there are, to which extent one can exploit or one can try to understand the qualitative different nature of the behavior of interactions and the reaction of some active colloids to the fact that they are out of equilibrium, not necessarily under external field. So depending on how far I go, where do I get, I wanted to focus on one particular case that is standard and that we control well on the lab, which is the use of colloids that are treated on their surface so that they are heterogeneous. They typically catalyze a reaction, and that by electrophoresis or by diffusophoresis allow them to propel. And then the question is, why do they do that? How do we should understand this, and how do they do collectively? If I have time, then there is a second part where I wanted then to talk about how this is how we directly exploit, understand the activity. The other is, if the environment is complex, is heterogeneous, how these particles interact with the environment and can also react or modify the environment and produce also certain structures, okay? So, as I said, this is a standard, it's a well-known, well-used, I think still very fundamentally there are questions to be understood, but really, so the basics is that these particles, because they are heterogeneous, the catalyzer, say the red part here, decomposes a reactant, and say what I will be showing you, this is motivated by experiments done with decomposition of oxygen peroxide in one case and by decomposing urea. So one can use platinum, for example, or there is a possibility. Now people, for example, also cover this with an enzyme, because then the colloids are more biocompatible. So for some of these biotechnological uses, I mean, you cannot do that with platinum and oxygen peroxide for obvious reasons. But at the end, one has a reactant that is being decomposed in one part of the colloid, the colloidal surface, and as a result, there will be gradients in reactants and products. If I don't enter, I mean, the details of the chemical reaction itself is non-trivial, but at, say, lowest order, or trying to understand mechanisms, one can think that there is a field phi that is this difference in reaction, in concentration between reactants and products that will not be homogeneous over the particle surface. And if these products are attracted to the colloid, then typically this gradient in concentration over the particle surface leads to a motion by phoresis. And phoresis is a classical subject in colloidal physics or in soft matter, and actually there is a nice review by Anderson that explains all these different mechanisms of phoresis, thermophoresis, diffuso or electrophoresis, how they can be understood on the same framework. And the basic idea, just to set the frame, is that if you think on the surface of the colloid, here being vertical, and the colloid attracts certain species, the concentration of these species perpendicular to the surface will not be uniform, will, I mean, in lowest order, will be an exponential profile that will depend on phi, the potential of attraction. So that's the physical chemistry of the interaction between the chemicals and the colloid. And here chemical potential is uniform, and the pressure also here will be uniform. But I mean, the pressure will be the hydrostatic pressure when we move perpendicular to the wall. But if I look up and down, if I put here a gradient, because here say there is more reactant that is being decomposed, these gradients along the wall means that even this prefactor will change along the wall, and as a result there will be also a change in pressure along the wall because of the local hydrostatic pressure. And if you now have a pressure imbalance by Stokes equation, then that will lead to a velocity profile. So this pressure imbalance generated by the concentration gradient leads to essentially a profile that will be characterized by a characteristic difference in the velocity away and the velocity at the wall, and that's the slip velocity. So that's the basic idea, and that presumes that there is this equilibrium. This is essentially what I write here, and this is typically what we use to understand why these colloids move. However, and I wanted to show this at the single particle level, then I will look what happens with many, but I think this is an interesting example of how subtle the fact that systems are out of equilibrium is, which is, and this is slightly more complicated than the case I've shown you before, and these are experiments done in Samuel Sanchez's lab in Barcelona. Essentially, he considered one of these colloids with this enzyme, and this enzyme, when it decomposes, the products have a charge, typically. So this is in water, so you have ions, which means then that, say, the black part of the colloid that is producing, where the chemical reaction takes place, where the decomposition of the corresponding molecule takes place, will lead to effectively a flux of cations and ions. So one can solve the equations in steady state, but because it's a constant flux, at the end of the day, this is a source. It's a source of ions, and if you have a source of ions that diffuse, actually the profile of the concentration decays as 1 over r. So this assumption that is local and we can forget about the flux that I mentioned in Anderson is conceptually not correct. It's a good approximation in some cases, but it's not correct. Now, in this particular situation, if you think like in these profiles that decay as 1 over r, if the diffusivity of the two ions is not the same, then the prefactor by which they decay, the rate at which they decay is not the same because it depends on the diffusion coefficient, which means then that the charge is not really localized in this Anderson double layer that I was telling you before. Charge spreads over the whole system. So then, even if the process is in essence that I was telling was diffusophoretic, there is this electrophoresis component that you cannot get rid of or can sort of like integrate out in the effective potential, as the Anderson picture would tell us. And this then, there is then, what we did was to look at this problem in general. There are, again, it's a fluid mechanical problem. I don't go into the detail. If you have questions, we can sort of go in, but it has no secret. Stokes equation, the diffusion advection equation for the species, and the boundary condition. So here the Cs is co-ions, the anions and cations, and they are produced as a given rate. So there is one dimensionless parameter that is the rate of the production with respect to how quick these ions diffuse. There is another parameter that is this asymmetry in the diffusivity, and actually, I mean, one do the derivation using the Lorentz theorem and so on. One can get to an expression for the velocity that depends on the charge of the colloid and depends on this ratio beta. The ratio, if beta is 1, essentially, I mean, then you will have what here is HOT is the standard Anderson prediction but then there are these two terms that have different parity in beta. And then that means that then again, when you look at values, so this is the velocity as a function of this rate. So if the rate is very low, essentially the red line here is Anderson, so it works. So all these details that the process that I'm describing quantitatively is really not important, but as the rate increases, the rate of production of ions, the activity increases, there can be quite a large, I mean, this is a logarithmic scale, so the difference in magnitude is quite substantial, and not only the magnitude, actually, depending then on the charge of the colloid, there can be cases where, I mean, this is a busy plot, but if you look, for example, here at the red points, then the velocity can change sign, which means that changing the rate of activity, not only the speed, but also the direction. So one can tune the directionality in that way, which is, again, something that in equilibrium or in the limit in which we are in a quasi-equilibrium state is not possible. Actually, if you want visually, what happens when we increase Q is that, I mean, in the low activity region, here the color, I mean, this is half a colloid, and this is the numerical solution of the equations that I was showing you. The color is the amount of salt. So greenish or bluish green is kind of like no charge. So here is really the charge accumulates very close to the surface. This is this double layer limit. That's the Anderson limit. And then as Q increases, what happens is that charge starts being decoupled. Numerically, it's never zero, but it's very, very small. It becomes being relevant outside this double layer, and that changes then there is an electrophoretic component that actually counterbalances the initial motion. That's why it can change sign. So that's for one, simply to say that one cannot take for granted that what we can know in equilibrium, we can just directly extrapolate. We need to understand what's going on to see when this standard description is valid or not. What I wanted to discuss now is what happens when we have a number of these colloids instead of one, but it's another experiment. It's motivated now by other experiments. So these are experiments that are covered where the active colloids are covered with platinum and they decompose oxygen peroxide. And here I will assume that we can use this Anderson picture. There is only one species and a charge is not taken into account. It's purely diffusophoretic. But here I wanted to look at another effect which shows how we can manipulate or make use of this, of the fact that these systems are producing these ions, this species out of equilibrium to generate and control how they attract and aggregate in this case and move. And this is an experiment that so basically what they consider in the experiment are these are the active particles, and these are sort of like micron, a bit more than micron size. They have some hematites out there. They tend to sediment. So at the end, what you will be seeing is a monolayer of these particles. So they sediment on a wall, and then we see what happens in the presence of the oxygen peroxide. Now, as it is drawn here, these active colloids that they work with are not perfectly spherical, and it turns out that when they sediment, they tend to sediment with the platinum pointing towards the wall. So basically, a single one of these active colloids will be sort of like decomposing oxygen and essentially sort of like hitting against the wall. Yeah? So you may think that this is a bit stupid because the particle is doing nothing. It's basically hitting the wall, and then because it fluctuates a bit, it will diffuse around. But again, this is producing, it's true, it's doing nothing, but it's decomposing oxygen peroxide. So there is a gradient of oxygen peroxide that, again, goes as 1 over R because it's a, it's a sink, if you want, of oxygen peroxide. So now if there is another particle that has a diffusophoretic response to the oxygen peroxide, we'll see a gradient, and then we'll move in that gradient. So then you can have an attraction that is essentially a collective effect, because isolated particles by themselves wouldn't move. You need a few of them. So this goes in the direction of looking into swarming, into how these particles collectively can respond or can behave because of their activity. So what they did is to mix these active particles with silica particles. The silica colloids are passive, but they have, because they also have a silica component. Both species react to the diffusophoretically to gradients in these oxygen peroxide. So if you have 2 of them, and this is experiments with 2 particles, one passive, one active, then what happens is that the relative position decreases. Sorry, that's the relative position, sorry, the blue one, and the red is the center of mass, I think, and then at some point it will start moving. So when they come together, then they can propel. So they form a dimer, but the dimer is not bound. So if we stop the oxygen peroxide, they will move away. So they are bound dynamically because of this interaction, which is persistent. So again, what we did is we could estimate what is the force that this gradient creates on the silica particle. Silica particle perturbs the concentration, so also affects how an active particle will move. This interaction is not reciprocal. It's not the same. The force that the silica particle distorting the chemical field affects the active particle is not the same than otherwise, but one can compute at the pairwise level the problem of at low Peclet numbers, what is the chemical fields, and integrating over the surface, assuming that the slip velocity is proportional to the gradient, identify these relative velocities, and then we can do now a Brownian dynamic simulations of this mixture of particles. We'll have active-active interaction, active-passive, and passive-passive. Okay? Yes? Sorry? Because of? I'm not sure what you mean by screening. So,
exhibit this.
Yes. So then each particle generates this long-range algebraically decaying field, and that leads to that algebraic interaction with power 2 or 5 when you solve the problem. Now, it's not exponential. That's what you mean? Or you refer at the exponent of the relative interaction?
Yeah, exponent not being 1 over R, but some other.
So the exponent is 2 and 5. Now, technically speaking, this is a problem. We solve this problem without the wall. Now, the wall can affect that exponent as well. We think that's because they are slightly away from the wall. We think that that's not fundamental. What we have done in practice is we have solved the problem for 2 spheres, and that's what comes out in the bulk. And then we have matched them with experimental data to fix the prefactors so that at least we can compute semi-quantitatively with experiments. But the wall can have an effect on but I would think it's always algebraic. I don't see a reason to having a true screening, because this is like a monolayer. You have enough of the reactant on top, so I don't expect that because they come together, then they deplete, for example, significantly the amount of oxygen peroxide that can affect their mobility. Okay. Yeah. So we can solve that, and then in the experiments what they do is, and then we can compare with experiments. So they have this monolayer. This is an image of the monolayer. The active particles are dark, and the silica particles are lighter. And then they actually, something that is useful from the experiment point of view is that the platinum, in principle, catalyzes the oxygen peroxide, but the efficiency with catalysis changes a lot when they are illuminated with the right wavelength. And in the experiments, they use that as a switch. So basically, here they have everything ready, and then they illuminate with the right wavelength. And then when they do that, it's quite quickly that they see how these particles approach each other, aggregate, and they aggregate by sort of like trying to, with a certain structure between the active particles and the passive ones. Yes, they can. But not by the wavelength, they do that by the intensity of the light. So, the wavelength is what is the switch, is that what is sensitive to, and then depending on the intensity, they can be faster or slower. And then basically what they did in the experiments is to vary the concentration of silica particles and the concentration of these active particles. And basically, if the density is weak, small, then you will have aggregates, clusters that move around. And there are regimes where you have these structures that basically span the system. These are like gels, and at very high concentrations is more like a glass. And as you can see here, if we have more active particles, these gels are like more elongated, while if we move more into the trying to get the equivalent one is a bit more difficult by increasing the silica. That's not the case. This is because these particles, these active particles, because they have some hematite, they also have a magnetic component. So when they come together and join, they tend to form slightly elongated structures. Maybe you could see this here. In the simulations, we also introduce this attraction for comparison, but that's not fundamental for what we see here. So we could identify, in the simulations, so these are the experiments, and then these are the simulations. We could sort of like more or less get the equivalent structures. So at the end, it's possible to get a phase diagram where we have different type of clusters or gels, and we call it gel because we look at the dynamic intermediate structure factor, and we saw sort of like a very low decaying plateau in the angular, in the size 6 order parameter, because this is a 2D system. So this tells us that indeed we can control and form structures by modifying the activity and relative composition. And I wanted to, how I am doing with time? Because I didn't pay attention. 30. Okay, perfect. So I can finish this and then still explain a bit of the other. I wanted then, so this is just to indicate how activity really, even if, as I said, even if individually they wouldn't do anything, collectively they are able to, or we can manipulate by using I didn't come into this, but it's true that changing the activity, you can also manipulate when these phases or when these different structures appear, and then that's a way to control them collectively without having to go one by one and see exactly what they do. And I wanted to show you also, this is to show that things are sometimes subtle. In the same simulations, experiments, in the same experiments, they also work in cases where the concentration of the active particles was very small. And in those conditions, basically, they could focus on individual clusters. So here you see the cluster, and this is an example of a different power. So with power, they favor that they are larger or not. So they change, so they can observe these clusters at different sizes, and they could measure for example, for the different sizes, the velocity decreases with size, and also if you look at one of them, the mean square displacement has a t square, so they at some point propel. This may seem, seemed a bit surprising, because in principle, I would have thought that these objects, these rafts are quite symmetric, so there is no need for the particle to move. But actually, when one starts looking into the shape, it's not perfectly a circle. And also on top of that, it turns out that when they looked a bit more in detail into the structure, the hematite, the active particles, was slightly off-center. And actually so they could measure I mean, this is exaggerated. It's not that off-center, but it's sufficiently off-center to make it displace. And there was a systematic dependence of this asymmetry with respect to the area. So we understood that this explains why they propel. So it's non-trivial that, I mean, in retrospect, you can think that if they start moving, because the particle that is active generates certain motion, it's slightly asymmetrical, the probability that other particles have to get to the cluster, and that we think creates that asymmetry. But something that we found a bit also, yes, sorry.
This cluster stable in the absence of light?
No. Everything is sustained by, I mean, it has to be active. So light is the way, so let's say, if it can decompose oxygen peroxide by other reasons, then it will be sustained. Light is the practical way to ensure that the rate of production or decomposition of oxygen peroxide is large enough for the interaction to be strong, to keep them together. But if you switch off the light, it's equilibrium, they will dissolve. Yes. So there is no strong attractive interactions between them, between these different colors.
I thought light is only adding as or used as some catalysis to the decomposition process.
Yeah. So let's say what I understood from the experiments is that in the absence of light, the efficiency of platinum as a catalyzer is so weak that the amount of material that did decompose, or the gradients that are generated, are too weak to really drive a significant attraction between the particles. That's what I understood. Yeah. That's why I said, I don't know if instead of platinum, you could put another or an enzyme that would be more efficient by itself, then you could see that without the light. Yeah. Something that, so as I said, so they had the experiments, and then we could compare with the simulations, the ones that the same model that I described before, and we could compare the power law, so that was working. But something that we observe is that in the simulations, the aggregates, the rafts that we observed, had a stronger tendency to change their motion. The persistence was smaller than in the experiments, and also when we look at where the particle was, when we start looking at the geometry, the structure of these aggregates, in our simulations, the active particle tended to be in front. So if I look in the direction which the raft is moving, was in the front, while in the experiments was in the rear. So that's something that puzzles us a bit, because we didn't know exactly why. So we look a bit more in detail, and because there is something that I have haven't paid attention to, which is hydrodynamics. As we heard in the previous talk, hydrodynamics is important. These are actually, I mean, the system is complicated. I mean, you were asking, there is the question of the wall that I was telling before already at the chemical level, from the chemical interaction, but actually I've been emphasizing that if there is a gradient in concentration, then because of this gradient there will be this diffuser-phoretic response, but because these particles are on a wall, then it means that if there is a gradient, and actually the material of the wall is the same as the material of the colloids, the wall itself also has a response to these gradients, which then is osmophoresis, gives rise to an osmotic flow. So in reality, we have both. We have the fact that the particle has this self-diffuser-phoretic due to the gradients that the particle generates, but also these gradients that the particle generates will lead to surface flows. So this raft that is on top of a wall will be subject to a certain flow below and above it. And we initially thought that that would be a second effect, in the sense that would simply renormalize whatever is the effective friction, but we decided to look into that, and then again, for that, then we have to say that we have to write down, and we did that theoretically, again, using the reciprocal, the Lorentz reciprocal theorem. We can say, well, we have an object that is force-free, so the force due to this friction plus the force of the force of the phoresis plus the drag have to balance out, and then you can write this down. It's a bit lengthy, but then one can have a prediction about how the velocity will depend on the particle the object size. In this case, we give an object size and see what happens. And we have actually two mobilities. We have, oh, it's here, yeah. We'll have in reaction to a concentration gradient, there will be a velocity on the solid surface characterized by the diffuse osmotic mobility, and because the particle will have a tendency to move in a gradient concentration with a diffusophoretic mobility, we have these two coefficients. And actually, because both flows respond to the same gradient, actually, this chi here is like the gradient with other terms. Actually, the velocity of this object depends on this gradient and depends actually on the difference between these two mobilities. So in a way, the phoretic flow and the osmotic flow have opposite directions. So this is simply telling us that because there is this competition, we can expect that since the solid wall and the colloid are made of the same material, both mobilities will have the same sign. So it's a question of which one is stronger. So the fact that it goes opposite to the direction that would assume the phoretic mobility suggests that the osmotic mobility of the substrate is higher, and that then determines that it's moving in the opposite direction. So that's told us that if we want to understand properly, not the kinetics, I think it's captured already with this phoresis, but if we want to capture properly how these rafts move and interact among themselves, then one cannot leave aside also the fact that they move and generate flows and interact also through these flows. So that's something to look in the future. And in the remaining sort of like 5 or 7 minutes, yeah? Now I wanted to change gears and then look at another aspect in how these active particles can interact collectively with a medium that is a bit more complicated than a solvent. A Newtonian solvent is always complicated, but this is a medium with a structure. And I will do that by, again, looking at simulations. In this case, there are no experiments so far. And I will also, for the time being, disregard hydrodynamics. So what we looked at, in principle, is something that is relatively straightforward. So we consider a binary mixture, a fluid binary mixture that can phase separate, in which we have active colloids. And we did that by assuming or considering that now the colloids behave as active Brownian particles. So they will move with a characteristic velocity that will depend on their orientation, and then there is a colloid-colloid or particle-particle interaction. They cannot overlap. And then they will also interact with the binary fluid. Actually, we do that by the binary fluid is simulated through cos-grain by a field psi that will be +1 in one phase, -1 in the other phase, and we solve essentially it's like a Ginzburg-Landau free energy, and we solve the Canhillier model for it. So this is a continuum field. Particles are individual particles, but each particle has a size. The field can go inside, and then we introduce a coupling free energy between the particle and this field. That's how it will prefer to be in one phase or in one liquid or the other. And then associated to this coupling free energy, there is a force. And in principle also, this direction of motion will diffuse around in 2 dimensions. It's characterized by an angle, so it will have a rotational diffusion. And also then there may be a coupling, in principle, between different colloids, between different particles. There could be an active torque that we will not introduce, and can also be a torque with the order parameter if it's not, if there are gradients there, okay? So all this comes from a free energy, so in a way it has a well-defined thermodynamic limit. And what we do is we solve this hybrid. So we solve in a lattice this order parameter, which is a field, and then in this field that is moving, at the same time then is coupled to these particles that are moving around. The motion, the level of activity of these colloids, well, because I mean, as I said, they self-propel at a given characteristic self-propelling velocity, and the inverse of the rotational diffusion is like time in which they sort of like lose the memory in which they are moving. We will define as usual, and it's usually done this active Peclet number, which is a ratio between these two times. So so a Peclet large means that the particle can displace long distance compared to its own size before it will rotate away. So what happens if I now take this binary mixture at high temperatures? Now I do a quench, and then so it starts from an initially homogeneous state, and then it will phase separate. And then if I look at the characteristic domain size, what I see, what I will be showing you is for a 20% area fraction of these suspended particles. Then typically it coarsens. It coarsens with a power of 1/3 if the particles are not very active. And at long times, you will have complete phase separation. I will be showing you results when the particles, the colloids, are soluble in the white phase. Okay? Again, that's controlled through that free energy I was mentioning before. Now, what we observe is that as we increase Peclet number, there is then a regime where the coarsening is arrested, and what we see is basically a state where the active particles have broken up the other phase into, this is for one particular case, into domains that are relatively spherical. So this is more like an emulsion, if you want. So active particles are able to emulsify a binary mixture. So this is 5 minutes. Okay. Yeah, okay, I'll try to finish. So in order to look a bit more in detail what's going on, rather than looking at this, we started with an initial condition where the two phases are completely phase separated. So now, if we have complete phase separation, the domain size, if a Peclet is small, keeps being large, so nothing happens. And indeed, as we increase activity, then the typical domain size decreases. We more or less end up in the same place. So what we see as the final state is, I think it's a reasonable, really well-defined steady state. And indeed, there are, so this at very low activity, the active particles stay in their phase. At intermediate Peclet numbers, then there are these domains. At even larger Peclet numbers, then there is another state where domains stay there, but they are smaller. And then eventually, when particles have enough activity, at the end, they can traverse across all interfaces, but then they end up accumulating in the wrong phase. That's the phase where they are not soluble, and this is essentially because in the phase that they are not soluble, because they have a stronger repulsion energy of forces, their actual velocity decreases, and active particles tend to accumulate in regions where their velocity is smaller. So they are trapped there dynamically in a way. So we computed the typical domain size, and as I said, in this region 1, this is the system size. Then we have this decay. Actually, the decay of the domains goes as 1 over the Peclet, and this can be understood if you think that what happens is that at the end, the binary mixture wants to phase separate, so there is, if you want, an energy associated to it that is because of the surface tension. And then, on the other hand, the active particles have an active pressure. So if we sort of like assume that this is the balance between these two pressures, the capillary pressure against the active pressure, actually the prediction is that the radius of the domains goes as 1 over Peclet number. So at least the scaling can be understood as a way of balancing these two. But maybe it's more clear what I say if I show you a few movies. So this is low Peclet, and yeah, you see that basically the domains stay being separated. When we go to these intermediate cases, then now activity is able to break up the gray phase into a state, an emulsion, where domains have a quite relatively well-defined size. You could even wonder whether they want to have a certain structure, order. And then at some point, so here essentially the active particles still stay mostly in the soluble phase. It's difficult for them to cross because there is the cost of the energy, the capillary energy. Now there is this intermediate, what I will show you, where they can go through start being able to go through the gray phase. And here, it's not just that the domains are smaller, it's that they are much more dynamic. You will see that now, so we classify this as 2 types of active emulsions. both are active and different from passive ones, but here is really more, here we don't have a balance that will tell us what's going on, but we see that particles can go in and out these gray domains. And now here, when they have a large enough activity, then essentially they go to the gray phase. They can go there because then they can surmount easily the capillary forces and then stay in that phase because they move slowly, more slowly there effectively. Okay, so I think that's an example of also how collectively, because that, again, you cannot do that at individual level, but this active pressure can create new types of structures, in this case, when you have a medium that is more complex than just a homogeneous medium. And that's, I don't know, I mean, it's something that is still, I don't know to which extent that's relevant or not, but there is this now a lot of interest in biophysics in understanding how inside cells one can sustain structures that are liquid where surface tensions are small, when these liquid-liquid phase separations that are arrested. And actually this shows that activity may play an important role in determining how these intermediate structures that are metastable, they are not equilibrium, can be sustained dynamically. And actually this shows that there are different regimes by which, or different mechanisms by which such type of emulsions can be sustained. So I think I'll finish here. There was now a last part showing that also activity can create spontaneous chirality, but I will try to skip that, sorry, because I have to go through all these movies, yes. Simply to say that I hope show you the importance of medium and the chemical interactions and how that can lead to new scenarios to manipulate these clusters dynamically, both in the case of this self-diffusion for colloids, or also in the case of the inhomogeneous media, how we can have or promote, stop these phase separations and generate new non-equilibrium structures. This has been done in Barcelona. The theory, I mean, most of the simulations by Javier Diaz, a postdoc, and Sergi Granados, a student, and as I mentioned, the experiments were done in the group of Pietro Tierno with Dola Chai and Elena. And thank you very much for your attention.
Thanks, Ignacio. Questions? Kabir.
So very interesting. So in the ABPs, when you have this growth of this length scale for different Peclet number, you seem to have a t to the 1/3 law, which corresponds to the Lifshitz sort of growth. But that's equilibrium, if I'm not mistaken.
Well, I mean, that's so I couldn't call it, this is coarsening.
Coarsening, yes.
Controlled by diffusion against interfacial energy in that sense. So yes, so it's equilibrium in that sense. And indeed, if you think, we see this coarsening when Peclet is not very large, or even when it's large initially, we see that probably because domains are small and can start growing, and then at some point it's arrested. So I would say the activity part of this pressure generated by the particles acts precisely at arresting what this underlying kinetic phenomenon.
And when does the crossover happen? That's governed by the active time scale.
Yes. So what I didn't put here the expression. So we have this estimate that we say that the arrest is controlled by a balance between the active pressure, and the active pressure is proportional essentially to the velocity and to the number of particles against the Laplace pressure. That sets a scale, and then that will tell you when you will be arrested. So the arrest is not a question of time, it's a question of length scale when these two pressures are balance each other. If the domains are very small, capillary pressure is large enough to favor growth, and then at some point the activity is able to arrest it.
So there's a critical droplet size, because the Laplace pressure for that size droplet will match.
Yeah.
I see.
Yeah.
Okay, thanks a lot. Yeah.
I have a quick question. So suppose the active particles had a tendency to flock, and now you put them into this complex environment. So have people thought about this?
I mean, no, not that I'm aware of. Actually, in the part that I didn't talk about vorticity, there we look at it because that's very important to promote chirality in confinement. But that's a natural follow-up to see now what. That's why I said I show you the simplest case, no? But that's the fact that this happens now. It definitely makes it relevant to try to see now how other aspects of activity influence, interact with this mixture. Yeah.
Thanks for a very interesting talk. So you showed us a phase diagram where you had 3 different kinds of gels, gel 1, 2, 3. So how do you differentiate between the 3 phases and which quantity is responsible to create a differentiation between the...
To go there, maybe it's why because I'm in the wrong... why can I come back here? Yes. Yeah. So, in short, these are related to the fact that the active particles are magnetic. So at the end, so what we call gel 1 and gel 2, I mean between 2 and 3, honestly, so how we distinguish? We have a measure about the asymmetry of these domains.
Okay.
So basically, let me see if I go to the next one. Yes, here we can see where gel 1, so gel 1 is here. That's what I say. These are gels where there is a majority of the silica component, while the others are gels where there is a majority of the active component. The active component, because of the magnetic interaction, has this elongated, they have a tendency to form these very elongated sort of like arms. So if you try to now measure a bit the dispersion along a characteristic length, one is more asymmetric than the other. Okay. It's a morphological measure to that. Between 2 and 3, honestly, I think the 3 is when we are moving in the active component, when we lose that, when the asymmetry becomes weaker, so it's a bit more isotropic, but I would need to look back again. I was always a bit doubtful about the 3. So 1 and 2 for me is clear, because of this different way to aggregate the particles, especially the active ones.
Thank you.
Vasa first, then Abhijit.
The last part of your talk, the initial configuration, if I understand correctly, you had a white phase and a gray phase, and white had active particles and the gray had passive? No particle.
No particle. Okay. So the active particles soluble. If the active particles are not active, then they are, anyway, thermodynamically, the particles are soluble in the white phase.
Okay.
And then, but all the particles are the same. All are active. And then when I change is the degree of activity of these particles.
Sure. Yeah. Thanks.
The active pressure is sort of dependent on the velocity, the activity.
Yeah.
What's the role of the length scale of the active particle or its mass?
What do you? So there is no mass. So this
in this
is
case,
Ovidam.
okay. There's no
in
inertia.
This case there is no mass.
Okay.
So there is a length
scale
which is the persistence length of the particles, but that's again encoded on this active velocity. They go together. They are not independent.
More questions?
That's another interesting direction, which is also an ongoing issue. That's a bit more formal, but the role of inertia on active particles, which then would be specifically the mass, is always something that tends to be left aside, and exactly when or how it applies, I mean, there are some works, but I don't think it's completely settled, so that could also be interesting.
Yeah.
Nice talk, Professor. So that last part you just flashed, that I was wondering that spontaneous chiral symmetry breaking. So will you please mind to just briefly say that phenomena, what you get there?
Well, just very briefly. So basically, what we, maybe I can just, if I have an image, I can tell you. So essentially, the idea is we have the same type of particles now on a phase which will form domains, spherical domains. We do that, I mean, this is for a block of polymer, but the point is that it's a soft spherical circular confinement. Now, if the active particles, if they are, in this case, they were soluble in the gray phase, activity by themselves will favor that they tend to go to this interface and stay there. What we saw is that, in this case, that's where we were introducing the alignment. If we introduce an alignment between the particles, when they come there, they have this tendency to move, to be sort of like perpendicular, but if by a fluctuation they sort of like start rotating or being aligned, moving, say, parallel to the wall, to the interface, then they have a stronger tendency to stay there for longer. So they still can come in and out, but statistically they tend to accumulate into what we call trains in an intermediate region of persistence, and that then gives a spontaneous
breaking because there is nothing that is chiral in the interactions themselves. Each domain can sort of like obviously rotate in different direction, and that's say how they create this vorticity.
Okay, I think that's the end of this year's Chennai Soft Matter. Thank you all for being here. It's a large number to be end with, which is good. So on a Saturday afternoon. Thanks to all the speakers, and especially those of you who have come from outside Chennai. Thanks to all of you for being here. And I would also like to thank our admin for facilitating the meeting, our housing, catering, media, and electrical team to help us out, student volunteers for different things, and of course my co-organizers, Basa and Arnab, for doing all the back-end work so that we can fix this meeting. So we see again next year sometime. So, and maybe we go back to IIT for the next meeting and so on. Okay, so thanks to all of you. There'll be tea and coffee and some snacks outside. Enjoy yourself, and maybe still some time to look at posters. Thank you.